I'm Dr. Alex Danis, and this is P51, the Molecular Fluorescence Viewer from miniPCR Bio. It might look like just a cardboard box, but if we flip this switch, I can show you it's actually way more than that. With P51, you can detect and measure molecules like DNA in a simple visual way. Scientists use fluorescence every day to detect and quantify DNA, RNA, and proteins, but it often requires sophisticated and complex equipment. With P51, we've packaged the power to visualize molecules into a compact, affordable, handheld format that's perfect for any setting. Using P51, students of all ages can directly visualize the fundamental molecules of life in action. For example, you can watch as DNA forms a double helix and then unwinds. Or you can see enzymes process a substrate. In doing so, P51 brings affordable, accessible activities to areas of the biology curriculum where there weren't any before. For example, our BioBits Central Dogma Lab allows you to explore protein production in real time. In our bodies, the information from DNA is transcribed into RNA, which codes for proteins. While this is happening all the time in your cells, you can't see it. But now, using the P51 together with our BioBits Central Dogma Lab, you can watch transcription and translation happen in real time. With our Beta-Gal Glow Lab, students can manipulate enzyme reaction rates using concentration, heat, pH, and competitive inhibition, all in an easy handheld format. A simple visual output allows students to graph reaction rates with a fast and easy setup. And while biology labs across the world use fluorescence to detect DNA in their samples, it often requires expensive equipment. But our DNA Glow Lab uses a fluorescent dye to detect double-stranded DNA cheaply and easily. And if you break the bonds of that DNA, the fluorescence will stop, allowing students to manipulate and visualize the structure of DNA in real time. So how does it work? It comes as a flat package that you get to build yourself, creating a tool that acts as a tube rack, a fluorescence viewer, and a tiny lab all in one. Inside are safe blue LEDs that illuminate your sample tubes. A special filter in the front lets only the fluorescence come through, revealing the molecules of life. It's simple, affordable, and easy to DIY, but durable and made to last. And right here under the lid is a picture of Dr. Rosalind Franklin. We chose to honor her because she helped create the first informative images of the DNA double helix, like Photo 51, that helped scientists model DNA's helical structure. Now, you too can visualize and examine the building blocks of life. With P51, you can make biology glow.